Hello students, now let us study embryo development, that too in a typical dicot. We start with the embryo development, but embryo development takes place after the development of the endosperm. Where is the embryo located? It is located inside the seed. What gave rise to the seed? Double fertilization gave rise to the seed. The ovule became the seed. Ovule or the megasporangium. Inside the ovule or megasporangium is present the embryo sac. Inside the embryo sac is the egg. An egg got fertilized by one of the male gametes and it gave rise to the zygote. This zygote will develop into the embryo and the triple fusion product, the primary endosperm nucleus, developed into the endosperm. Now we have two types of seeds, dicots and monocots, in which the two cotyledons are there. We call those seeds as dicots, in which there is only one cotyledon. We call that as monocot seed. But on the presence of endosperm, whether the endosperm is present in the seed or not present in the seed, we have two types of seeds, endospermic and non-endospermic. Endospermic seeds are also called albuminous seeds and non-endospermic are also called ex-albuminous seeds. Now this is the first seed, the monocot seed, which is endospermic seed. You can see well-developed endosperm here and a single cotyledon, the scutellum. This is a monocot seed. Where is the embryo? So here is the embryo. The one cotyledon, the scutellum. Inside that you can see here the plumule, which is being provided with a protective covering called the coleoptile, and radicle, which is being provided by a covering called coleorhiza. So we have coleorhiza, coleoptile, and the cotyledon. This is the embryo which is present in the monocot endospermic seed. That is the main seed. The radicle will give rise to the root, but natural, and the plumule will give rise to the shoot. Now the second seed is non-endospermic dicotyledonous seed, and it is that of a gram, the sisal. And you can see here the two cotyledons and the embryo, the plumule and the radicle here. So we don't have an endosperm here. So it is called non-endospermic dicot seed. We have a dicot seed with an endosperm, well-developed endosperm. And the example is Ricinus communis. You can see here the castor seed. The outermost covering is the testa, and just down below is the caruncle. And you can see here that inside you have got the inner covering. Inner to the testa is the perisperm, and you have got a well-developed endosperm here. And just when you remove the endosperm, you can see here the two papery cotyledons here. And where is the embryo? The two cotyledons and here it is the radicle and the plumule you can see here in the picture. So we have two types of seeds now, endospermic and non-endospermic. And in our syllabus, we have to study only a typical dicot seed, non-endospermic one. Now let us study the development of embryo. The development of non-endospermic dicotyledonous seed. The example is that of embryo of Capsula bursa variety pastoris. It is commonly called shepherd's purse. Development of the embryo begins after the development of endosperm. It's already clear. The diploid zygote divides and redivides to form an embryo. Now let us start with the development of the embryo, which takes place after the formation of the endosperm. In this case, no endosperm formation is taking place. This type of embryo development is also called crucifer type of embryo development because Capsula bursa variety pastoris belongs to crucifery, which is now called brassicaceae. Let us start with the zygote, which formed as a result of fusion of one of the male gametes with the female gametes because of syngamy. So this zygote is diploid. It undergoes mitotic division, giving rise to two cells transversely, Ca and Cb. Ca further divides vertically, giving rise to two cells, and Cb divides transversely to giving rise to Ci and Cm. That is, inverted T-shaped embryo, proembryo is formed. The two cells of the proembryo further divide to give rise to 4 cells, and Ci and Cm also divide to give rise to 6 to 8 cells. This stage is followed by, proembryo stage is followed by quadrant stage. Quadrant stage is followed by octant stage. These 4 cells give rise to 8 cells, and the 6 to 8 cells which are formed as a result of division of the Ci and Cm, they give rise to the V-shaped haustorial cell and 5 to 6 suspensor cells. So a suspensor is formed and a haustorial cell is formed. Haustorial cell helps in getting attachment to the of the embryo to the embryo sac and deriving nourishment for the developing embryo from the embryo sac. After the octant stage, the cells of the octant multiply periclinally. You can see here the periclinal division, shown periclinal division. So periclinally it will multiply and keep on multiplying. And the lowermost cell of the suspensor becomes prominent. Its nucleus becomes quite prominent and the cytoplasm becomes dense. This cell is called hypophysis. This stage is followed by the multiplication of this periclinal division. Not many periclinal divisions take place and give rise to a circular, somewhat rounded structure. And also the hypophysis cell multiplies. It divides to give rise to two cells. As such, we get a structure which is quite rounded and suspensor cells you can see here, and the ostoreal cell is also visible. This stage of embryo, since it's rounded in shape, since it is globose in shape, since it's globular in shape, we call this stage of embryo as globose stage. After the globose stage of embryo development, further divisions take place in embryo proper, especially at the points, at the 2 points in the lower tier. They are here somewhere, which are destined to form cotyledons. Now these two will be, you can see the formation of these two structures. These will now form the cotyledons. Embryo becomes heart-shaped. You can see here it's moving in. Here the invagination is taking place. It's moving in. So you can see that embryo becomes now caudate, or what we call as the heart-shaped in LS. You can see it is present inside the megagametophyte only. It's present inside the ovule only. The space is restricted. So what to do? They will keep on elongating till a horseshoe-shaped embryo is formed. Now here what happens, that since the space is less, after some time it will elongate till it occupies the whole space, and then the bending of the embryo will take place. So globose and embryo formation. Globose ultimately changes into a caudate shape, then it elongates into a horseshoe shape, and ultimately it starts bending. Embryo becomes heart-shaped, then after the bending will take place, the hypocotyle as well as the cotyledons soon elongate in size, mostly by transverse divisions of the cells. Bending of embryo takes place. I told you there is hardly any space, so embryo starts bending. Gradually, the cotyledons curve in such a manner that hypocotyle lies parallel to the cotyledons in mature embryo. You can see here hypocotyle, the region just below the cotyledons is called hypocotyle. Between the two cotyledons, there is meristematic stem tip, you can say tissue, which gives rise to the stem tip or the plumule proper. So here you can show, I'm showing with the help of the cursor. All right. And you can also see now what happens. The root tip and the root cap calyptrogen is formed from the cell called hypophysis. Now the stem tip you have seen between the two cotyledons is formed. There is a meristematic tissue, so which gives rise to the stem tip, and already some of the stages in differentiation of dermatogen, peribellum and pleroma are seen. You can see here this, the outermost is dermatogen, inner to this is the peribellum, and inner to it is the here you can see just below the radical, or this particular area just below the calyptrogen is the in the hypocotyle region you can find here the plerome. The plerome, now what happens to this? The differentiation has taken place. In future, the dermatogen will give rise to the root epidermis, and the peribellum will give rise to the cortex, and plerome will give rise to the conducting tissue, the xylem and the phloem. All right. So after this, the bending, you can see the bending of the cotyledons here. You can see here how the cotyledons are bending, and they are elongating also till they will occupy the whole space. Some of the stages in differentiation of dermatogen, peribellum and plerome you have already seen. Dermatogen will become the future epidermis, peribellum will become the future cortex, and plerome will become the conducting tissue in, because the first root formation will take place and shoot will be, it will be continued in the shoot throughout the plant. All these stages are seen in VS of vertical section of the seed of Capsula bursa variety pastoris. All right, you can see the mature embryo now. How does it look like? The mature embryo looks like this. The whole space in the ovule is now filled with two cotyledons, hypocotyle and calyptrogen, the root cap, and the radical here, and the plumule here. So whole thing gets covered by, you can see here the two cotyledons here. Alright, so that is the end of the mature formation of dicotyledonous embryo in Capsula bursa variety pet stories. So at the end, a mature embryo was formed, which actually comprises now of cotyledons and radicle and phloem. Suspensor vanishes. The hostorial cell is no more to be seen in the mature embryo and in the mature seed, and only the embryo can be seen. With this, we complete the development of the embryo in dicot, typical dicot, and the example we have taken of that of Capsula bursa variety pastoris. And last but not least, I would like to thank you for patiently listening to my lecture. And if you like it, share it, and subscribe. Thank you.